The nervous system, folks, that the neuron is pretty much the functional unit of the system, right? The neuron. You think about the neuron, it's a cell or the neuron, or they also call it it's a nerve cell, right? Nerve cell, okay? There are about 10 billion of them in terms of numbers. They are complex. They vary in size, and they have three functional parts to them. Okay, you have a body, or they also call it the soma, right? Or they also call it another term that describes the body of the neuron, nerve cell, is called a perikaryon. That's another. So all these terms, guys, perikaryon, soma, or simply nerve cell body, all describe the body of the neuron. Okay. Then you have you have what's called the input region, right? And guys, most likely the input region is going to be what? It's going to be sensory. Okay, the part sensory, and then you have the part of the conducting part, the part that's going to send the nerve impulse, right, the signal. So you have input part, the part that sends out the signal, or the conducting component, and then you have the secretory component of the neuron. Now what's going to be secreted from the neuron is very important. You have to think of a means of communication between neurons to another neuron, or neuron to a motor cell, and we call those what? Somebody said it, very good, neurotransmitters. Okay, what's going to be secreted from a given neuron to another neuron or whatnot is going to be the so-called neurotransmitters. Okay, and I'll give you a little quick overview of how that works. Okay, all right, so that's the overview of what encompasses the neuron or the nerve cell. You have 10 million of them. They're complex, they vary in size, the components of it, and what's secreted in terms of the circuitry component of the neuron. Now, folks, there are three categories. You have sensory neurons, you have motor neurons, and you have the interneurons, okay? Now, the sensory neuron is pretty much the receiver or the receptor that's going to carry the information back to the CNS. Guys, what's CNS? Central nervous system, okay? Now, that information is carried back or transmitted back to the CNS via, you have SA and VA fibers. Guys, what does SA stand for? S stands for somatic, right? Somatic or body. And A stands for what? Afferent, exactly. Okay, it's going back. Okay, well, if something goes back to a cell, you call that afferent. If it leaves from the cell, you call it efferent. Okay, so you have somatic afferent fibers. V stands for visceral. Very good. Visceral afferent fibers. Okay. So in other words, guys, if you get your finger cut in a door, if you step on a nail, if somebody pinches you, things of that nature. The fibers that perceive that information are your what? Your somatic afferent fibers, okay? Fibers that pretty much are distributed throughout parts of the body, general head, neck, so forth, etc. Somatic, okay? Afferent because it goes back to the CNS, okay? Somatic afferent fibers, you're able to perceive pain or those sensations, okay? Now, visceral afferents, this coincides with viscera, okay? Internal organs. So this coincides. So for example, if you have a tummy ache, I don't feel good. Okay, what's going to perceive that? What's going to carry that information? Relay it back to the CNS, the visceral what? Afferent fibers. If you feel a pain in your heart, if you feel a pain in your lungs, okay, what's going to perceive that? Visceral afferent fibers. Okay. So anything that coincides with being visceral, something that's visceral or a gut feeling, that coincides with the fibers picking up that information, sensations are being transmitted via the visceral afferent fibers. Sensation that you feel, head, extremities, and so forth, face, nose, mouth, etc. Those are somatic afferents. Okay. Then motor, it's pretty much from the CNS or ganglia to the target cell to the effector cells. And what sends that signal are the SE and VE. S stands for what, Ken? What, guys? Somatic. And V stands for what? Visceral. Okay. And E stands for what? Efferent. That is opposite afferent. Okay. So pretty much, guys, Afferent is, always think in terms of afferent being what? Sensory, efferent being what? The motor, okay? So, think in terms of, so in other words, guys, think in terms of you stepped on a nail or you get pricked by something or you get your hand cut at the door, do you just leave it there? Does it stay there? No, there's a kind of a reaction. You respond to that, right? There's a reflex to that, right? Your body moves, so what's sending that signal to your body to retract from that, from the source of pain, are your what? Somatic efferent fibers, okay? Now, visceral efferent fibers are going to be the signaling to the visceral organs. So in other words, ever heard of this right here, vasoconstriction? What is that, vasoconstriction? What is that? When your blood vessels, right, do what? When there's what do you call it? When there is a constriction, right, of the blood vessels, the walls of the blood vessels, narrowing the lumen, right? Okay, that's wired to the autonomic nervous system. Do you have control of your blood vessels doing this? No, you don't. Do you have control of peristalsis in your gut tube? No, you don't. That's all wired to your autonomic nervous system, right? But those are examples, or what does the mechanism of controlling the action or the effector or the motor of anything that's visceral would be your what fibers, guys? Your visceral what? Efferent. When you start to sweat, guys, ever heard of this? Ever heard of this? Oh, okay, like hopefully nobody will experience it. I mean, look, guys, if you do your homework, if you take notes, if you do what? What's the key word to this course, guys? I can't emphasize that. If you transcribe, right, you rewrite everything on a timely basis, not the day of the test, not 10 minutes before the test, you do it well in advance, then you won't be coming in here with that. Ever heard of this? Ever heard of that? Nervous sweat, like, nervous sweating? Well, what's causing that? What's causing you to sweat profusely? What's causing you to, your glands to, to secrete all that stuff are the what fibers? The visceral what? Efferent fibers. Okay? Do you have control of that? No, you don't. Okay? All right, so you have your sensory, what's motor, and then you have your interneurons, and that's pretty much the network that integrates the information that's in between. Okay? All right, then guys, here's the start of the show. Here's the topic of discussion, the neuron, guys, and again, here's the body of it, the body or the soma, or what else do we call it? Perikaryon. Okay? And then here's what sends the signal out, the axon, and this is what the so-called dendrites. We'll get to more detail on that in a bit, okay? All right, now guys, the neurons, they vary. You have, you have varieties of neurons in the body. You have the bipolars, right? You have the unipolars. I think your book calls it, I'm quite sure, either unipolar or your book calls it pseudo-unipolar. Both these terms are interchangeable, so if it's unipolar, pseudo-unipolar, it's describing these kind of neurons. And then you have the most common, guys. The most common of the neurons are the what? The multipolar. Make up 99% of the neuron population, okay? So the bipolars, here's a description of it, okay? Now these are more rare and you find them in your special senses, okay? In terms of, this is for vision and whatnot, okay? These are very rare. Then you have the unipolars. These are your dorsal root ganglion, pretty much, okay? And these are the sensory neurons of the what? What does PNS stand for? Peripheral nervous system, okay? Peripheral nervous system. So these are the pseudo-unipolars. And then the most common being the motor multipolars. These are, so pretty much guys, all your skeletal muscle motor neurons are the ones that initiate, movement of your muscles are pretty much the multipolar neurons. And they have myelinated axons. But we'll get to this in a bit when we start to talk about what is myelinated and what is unmyelinated, okay? So here are the varieties of neurons, and then here's a diagram showing you what they look like. You have this picture, guys? Okay. So again, here's the unipolar neurons, what it looks like. Here's the cell body, right? And then here's the axon, so forth, and here's the dendrites, okay? Then you have the bipolar neuron, and here's the body, here's the axon over here, here's the dendrite over here, and then you have the most common, okay? Just always keep in mind that this is the most common type of neuron, the multipolar one, okay? Now, the cell body, folks, it's got processes, or they also call these things neurites. And what are the processes with regards to the neuron, right? You have an axon, and you have your what? your dendrites, okay? So the processes include, or they call them neurites. They include the axon and the what? And the dendrites, okay? Think of dendrites as like little branchings, okay? That they normally branch, right? Now, guys, this is important, okay? When you're addressing nerve cell body, the soma, all right? Especially when we're talking about the central nervous system, aggregates, that's the term. When you have clusters, right, or aggregates, right, or just simply, clusters, right, of nerve cell bodies in the CNS, what do you call them? They're called nuclei, okay? And guys, this will come back to fruition. This will come back and haunt you when we start venturing into the brain, into the spinal cord, things like that, and then you hear the term nuclei. You can't think of a nucleus as the center of at the core of a cell. Okay, nuclei refers to, especially in the CNS, in neuroscience, nuclei is a term that they use to address a bunch of what? Nerve cell bodies in the central nervous system. Okay. Now, a bunch of nerve cell bodies in the what? In the PNS, what do you call those? Call them ganglia. Okay, so a bunch of nerve cell bodies in the PNS, you call them ganglia. Again, aggregates or clusters of these in the PNS, we call them ganglia. But in the CNS, we call them nuclei. Okay, so these terms are gonna again make sure that you get this in your vocab when you're addressing structures in this course, okay, or neuroscience in general. Okay, so cell body. Now, in terms of the processes, right, you have an axon, you have a dendrite. So the axon arises from the hillock. Guys, what the hillock is simply a narrowing. So here's the nerve cell body right here, right? But notice how it narrows. This part right here is called the axon hillock, right here. That's your hillock, very obvious. And then the long axon is considered a nerve fiber. Each neuron has one axon. The motor neuron impulse, or the so-called action potential, is initiated at the hillock. That's where it starts, okay? We'll talk about the action potential in a bit, in terms of, membrane potential, so forth in a bit. But the motor neuron impulse is triggered or initiated at the so-called axon hillock, okay? Where that part of the neuron, the body narrows down and it extends projects into the axon. Guys, what is the terminal? The terminal is it represents the secretory component of the neuron, but it is the terminus or the terminal of the axon. It is the end of the axon. So axons end and what are known as terminals. Okay, at terminals. What is axolemma? Axolemma is a fancy term that describes the what? The plasma, membrane of the what? Of the axon, okay? All right, guys, the axon is an extension of the actual neuron. Yes, it is, but since these things can travel great distances, they address the plasma membrane of that axon as the axolemma, okay? And we'll talk about that in a bit when we start to talk about the membrane potential, when you're talking about the action potential or the depolarization and so forth. Okay, but anyhow, but just keep this in mind. The axolemma, that term describes the plasma membrane of the actual axon. Okay, and then the terminus of the axon is the part where there is release or secretion of the neurotransmitter. So what happens, guys, is something like this. So you have a neuron, right? Here's a neuron. Here's the axon. Then it ends at what's called as a terminal. Here's a terminal, okay? And that's the part of the axon where there is what going on? Secretion. What is going to be secreted at this terminal, guys? The neural what? The neural what? Neurotransmitter. Exactly, the neurotransmitter is going to be released at the terminal, okay? So you have these little circles represent the vesicles that carry the neurotransmitter. What happens is, guys, and that's going to fuse to the membrane, right, of that terminal, and then what? Release the neurotransmitter into the space called a cleft. Okay? Anyhow, but we'll get to that in a bit when we talk about different types of synapses, okay? All right, so you have your axon, right? And again, guys, here's a soma, right? Here's a soma, or the perikaryon of the neuron. Here's the axon right here. That's the axon, right? Here's the hillock, here's the axon. This is the plasma membrane, right, of the axon called the axolemma. Well, actually, I should make it like this. You know, maybe do this. Maybe kind of like highlight the axon like this. The axon, and then the actual line is the axolemma. What is that? That's the plasma membrane of the axon. Okay. All right, and then, folks, then here's another picture, I think, showing you the axon, what it looks like. So here's the neuron right here. There's the narrowing of that nerve cell body leading into the axon. And again, here's the nerve cell body here, and then here's the narrowing of that with the hillock, and then you have extension of that, and that is the axon. Okay, so enough of axons. Now let's talk about the dendrites. Okay, they are short, they're tapering, branching, they have a lot of branches, okay? Guys, these are the receptors, okay? This is the part that receives information, or the what? The part that is what? Sensory info, receives sensory information, okay? Sensory part of the neuron. And then you have what's called the dendritic spine, guys. The spine is the point of synapse. Guys, when you think of a station with another neuron, okay? Synapse, it's like a space connection between neuron to another neuron, or neuron to a motor cell, muscle cell, I should say, muscle fiber. Okay. So let's take a look at that, guys. And here's a picture again. So here's the nerve cell body. We know here's the axon, but all these right here, all these. Okay, guys. So we have dendrites. We have what? Axons, right? Axons, dendrites. Okay. Now, guys, this information here addresses transport of substances within the axon, right? Axon transport. You have stuff, you have anterograde versus what is known as what? Retrograde. Guys, anterograde, think in terms of movement of substances within the axon from soma to what? To terminus. Okay, from the soma, nerve cell body to terminus, that direction is known as anterograde. Okay, from the soma to the terminus, that's anterograde transport. You have fast, you have slow. Now guys, what travels in terms of fast anterograde transport? You proteins are going to transport in there, and neurotransmitter, substances are going to travel rather quickly. And we're talking about, oh gosh, I don't know, about 200 to about 400 millimeters, right? Millimeters, right? Per day, I guess, per day, okay? So transports proteins and neurotransmitter substances. Now, in terms of slow anterograde transport, what moves slow, folks, and obviously from soma to terminus, is the axoplasm. Guys, what is axoplasm? That's a fancy term that describes the what? Cytoplasm of the what? Of the axon, exactly, of the axon. Okay. You guys recall the term cytoplasm, that's all the stuff inside a given cell. So the axoplasm is the cytoplasm of the axon, and that travels rather slow, about 1 to 3 millimeters per day. Very slow per day. Is that what it is? Yeah, per day. Okay? Just giving you some numbers, just ready to speak. Guys, the bottom line, you have fast and you have slow, okay? Then you have what's called retrograde. Now, guys, retrograde is which direction? From terminus to what? Back to the soma, okay? From terminus back to the soma. And what travels in terms of retrograde transport? You have vesicles that are taken in by pinocytosis. What is pinocytosis? That's a fancy term that describes cell what? Remember this from your bio courses? Cell what? We can call this, that's a fancy, pinocytosis is cell drinking. It's when a cell takes in fluids, okay, and they take them into these little vesicles, right, and they carry back to the body. So whatever that the cell takes in terms of fluids or cell drinking, that travels via retrograde transport. And also worn out organelles, whatever is, going to be recycled or not used or broken down, that travels from the terminus back to the nerve cell body, okay? Now folks, what's important, what I want you to get out of this is make you aware is that retrograde transport is also how things work their way back to the CNS. Okay, ever heard of this, folks? Ever heard of this? Rabies? What is that? It's a virus, okay? How do you get that? I don't know. You go out there. I don't think this campus, you see them around here, but who's taking classes up in BBC campus? Okay, well, you know what I'm talking about, right? Up there, especially if you go out late at night, and then you go out there, and then you'll see like a, I don't know, raccoon, right? Running around, maybe see some wildlife. Who's seen one? I remember I saw one years ago. Okay, well, exactly, you see them there, but guys, you know what? Don't feed those guys. Don't feed them. I know you may be, oh, but tree hugger, oh, how cute, yeah, you can feed them. All right, good luck. I mean, you'll feed them, but they'll take your food and probably what also? I bite your hand, thank you. And, well, that's the way of giving you thanks. I bite your hand, you're like, oh, okay. But are you okay with that? Getting bit on the hand? I don't think so, folks, because these animals harbor rabies, and that's a virus, their saliva, and how's that virus going to work its way back to your central nervous system? Through retrograde transport, okay? Folks, is that good? Oh, I mean, you get bit on the hand, you'll recover from your little wound, but folks, the time's ticking now. Now that virus is working its way back to your CNS and cause major problems via retrograde transport. Okay, so just folks, the moral story is if you see a raccoon out there and you're eating something, what do? Finish your meal and run away. Okay, just to stay clear of that critter and you'll be good to go. Okay, do not put retrograde transport to the test. Don't do that. Okay, because you're not gonna be happy. Okay. All right. What's next, guys? Now, neurotransmitters. You have several types of. I just give you some examples. You have acetylcholine. That's considered an excitatory neurotransmitter. Okay, it's a neurotransmitter that's obviously where you have a transmitter going from a neuron, right, to a skeletal muscle or muscle fiber, right? Then you have these so-called biogenic amines, which include these known as catecholamines. Catecholamines. And guys, the catecholamines include, norepinephrine, right? It includes epinephrine, norepinephrine. The older terms for these things are adrenaline and noradrenaline. You might have heard adrenaline, right? It's the same thing as epinephrine, okay? They are synonymous. I think adrenaline and noradrenaline is the older term. So examples of catecholamines include norepinephrine, epinephrine, and another one that's a catecholamine is dopamine. Okay, so I'm going to circle what are the catecholamines: norepinephrine, epinephrine, and dopamine. Okay, so those are the so-called catecholamines. I'm going to bring this up again when we start to talk about how these things function in terms of uptake or in the synapse. Okay. Now, guys, E or I, this stands for what? Excitatory. I stands for what? Inhibitory. Okay. So norepinephrine is excitatory or inhibitory. Dopamine is excitatory or inhibitory. Yes, serotonin. Okay, that's pretty much definitely inhibitory. It's involved in emotional behavior and your biological clock. And then you have amino acids type of neurotransmitters. You have GABA, which is inhibitor. You have glutamate, that's excitatory. And then you have glycine, that's inhibitory. Okay. So I just given you some very basic, just a small number of the so-called neurotransmitters for you to worry about. Okay. Now, guys, the synapse. Okay, you have two types. You have electrical versus what? Versus chemical. Okay. Guys, these are more rare. Okay, these are more rare. They're very rapid. They're less common. Their means of communication are known as via these cell adhesions. This is a type of cell adhesion, means of communication between neighboring cells, known as gap junctions. That's how the signal gets carried from one cell to the other. You find these in your heart cells, cardiocytes, okay? That's a signaling, how it goes from one cardiocyte to the other. And eventually they're replaced with chemical synapse. Now, in terms of chemical synapse, what you have is a presynaptic terminal, right? Then you have the vesicles, right, that fuse. Vesicles that, okay, first of all, guys, these vesicles carry what? Neurotransmitters, right? They fuse to the presynaptic terminal. Then you have a cleft, which is simply a fancy term that describes a little what? A space or the gap, right? And then you have the postsynaptic membrane that's going to have what? A kind of receptor. Okay, guys, so let's illustrate this real quick. So again, here's a nerve cell body, right? Here's your neuron. Here's a soma. Here's a nucleus, right? Here's the axon. Remember, it ends where, guys? At a what? At a terminal, okay. So this right here, then you have your middle vesicles, right, that travel in here, within the axon, and they carry the what, guys? The neurotransmitter, right? So the presynaptic terminal, right, is going to be this terminal right here, okay? So this vesicle is going to fuse to the presynaptic terminal, or presynaptic being before the synapse, it's going to fuse to the presynaptic terminal, release its contents, right? And then you have the cleft, the space, the gap, and then you have the postsynaptic membrane. It's going to receive the signal. Like guys, this membrane could either be a muscle cell, the plasma membrane of a muscle cell, or it could be another neuron, right? It could be an axon, a dendrite, and so forth, okay? And the postsynaptic membrane is going to have receptors. They're going to be here in the postsynaptic membrane, okay? And guys, these receptors are going to catch the what? They're going to catch the what, guys? Or receive the what? The neurotransmitter, okay? You have receptors on that membrane, postsynaptic membrane, that are going to receive or grab, snag that neurotransmitter, okay? So you have presynaptic terminal here, you have your cleft, your space here, and then you have your postsynaptic membrane, okay? Now guys, here's three types of, now in terms of synapses, guys, you have terms like axodendritic. What does this mean? You have a contact between an axon and a what? And a dendrite. You have one between the axon and what? The soma of another neuron of a nerve cell body, and then you have an axon to a what? To another axon. Okay. So let's take a look here, guys. These are pictures from your book. So, what would you call this synapse? What would you call this one here? Axosomatic. Okay. So it's safe to call that one as axosomatic. Okay. What would you call this one here? What do you call that one? Axo what? Axo what? Yeah, axoaxonic, very good, yeah. It goes from one axon to another axon. Okay, there's a contact there. What would you call this one? Axo what? Somebody said it, axo what? Somatic, yeah, look, it's from an axon to what? To a nerve cell body, so it's safe to call that one axosomatic also, right here, okay. What would you call this one here? Axodendritic, yeah, from an axon to a dendrite, okay? So, just guys, just by visual, just by looking at the diagram, guys, you can actually pretty much map out these types of synapses. Okay? It looks confusing, but it's not, guys. You just simply follow the axon and see what it's making contact with. The soma, you call it axosomatic. If it makes contact with another axon, axoaxonic. If it makes contact with a dendrite, axodendritic, okay? All right. Now, guys, this picture, are you okay there? Hello? You okay there? Confusion? You got it? Yeah, look, yeah. Look, I'm just saying, just all you gotta do is just, oops, or whatever. Just map out, just follow the axon. See, remember, it's always axon first, right? And just follow where it ends, okay? So this was dendrite, well, hang on. So you saw there were actually two axosomatics. You got axosomatic here, and another one where? Another one here also, okay? It's from the axon to the body. Okay, now guys, here you have, here, this is what happens with acetylcholine, guys. Acetylcholine is a type of neurotransmitter, right, that you see mostly, acetylcholine is a neurotransmitter that occurs at the myoneural junction. What does myoneural junction mean, guys? Myoneural. What does that mean? Exactly. It's a connection between an axon or a neuron and what? And a muscle. Okay, myoneural junction. See, acetylcholine, that's a neurotransmitter. Guys, look here. You have see little dots. The dots represent the neurotransmitter, right? Inside the vesicle. And notice how, what happens, guys? Remember, here's the presynaptic terminal, right? It fuses with the membrane, and then the neurotransmitter is released into the what? Into the cleft, right? And then here's the what? Postsynaptic, here's the postsynaptic what, guys? Membrane, right? Now, in this instance, guys, there's going to be what? Here at this membrane, a receptor, okay? A receptor, you can find a receptor, and it also has what? An enzyme. Guys, what's that enzyme going to do to that receptor? I mean, to that neurotransmitter? Get rid of it, clean it out for the next neurotransmitter. Okay, so you have receptors that have an enzyme. Okay, here. So here you have receptors on the membrane, right? On the postsynaptic membrane that's going to what? Capture that neurotransmitter, and then activate an active potential here or signal, and then get rid of what? Empty out that. Guys, think of it as like somebody sitting in that seat and then emptying out the seat. Okay, so you have an enzyme that's going to remove that neurotransmitter. And guys, every time you hear this, an enzyme always has a suffix ending what? ASE. Okay, so the enzyme that you're gonna find here is acetylcholinesterase. Okay, guys, bottom line here is that when it comes to acetylcholine, it involves some kind of receptor and what? And an enzyme to get rid of that neurotransmitter. Okay, clean it out for the next signal. Now, guys, here, this is for the catecholamines, right? For this one, what's happening here? You're gonna have what's known as reuptake, okay? Or just simply, guys, recycling. Okay, reuptake or recycling. So what happens is, guys, look here. Here's a neurotransmitter, right, in the vesicle. It's going to fuse to the membrane. It's going to release the neurotransmitter, okay? And then the neurotransmitter is going to be reuptaken by the same what? Presynaptic, right, terminal. It's going to be reuptaken again or recycled again, and what? And put to use again, okay? But this only applies, guys, to the what? To the catecholamines, okay? Acetylcholine requires a receptor, well, second receptor, but it requires pretty much an enzyme that's going to break it down and then to empty out that receptor to accept the neurotransmitter for that. But when it comes to catecholamines, it uses this method of reuptake, okay? So make sure you're aware of that. Catecholamines do reuptake. Acetylcholine uses a what? Okay, an enzyme to break it down, okay? Just very simple comparison of two types of neurotransmitters in terms of synapses, okay? All right, guys, then you have what's called the myelin sheath, okay? Protects and insulates the fibers, right? What does it do to the impulse? Guys, what does hasten mean? Speed it up, okay, guys? And guys, here's the rate of speed of the signal traveling along a myelinated sheath. It's 150 meters per second. Versus unmyelinated, less than a meter per second. Okay, so obviously the myelin sheath is important in what? Speeding up the impulse. Very important in speeding up the impulse. Okay? Alright, so guys, then the neuron has a nucleus, right? It is transparent, it is round, right? It functions as the biosynthetic center, right? It produces substances. It controls the manufacturing of what? Of proteins, right? It has a very conspicuous nucleolus, at the core of that nucleus, you'll see a little nucleolus, right? Very obvious. So if you look here, guys, here's a picture showing you a nucleus. So guys, here's the nucleus right here, and notice how it is transparent. You can actually spot out the what? What's this here? The nucleolus, okay? The nucleolus within the actual nucleus. So it's transparent, it's round shaped, and you find a very conspicuous nucleolus within the nucleus of the neuron, okay? What else, guys? Then you have the cytoplasm of the neuron. What does EM stand for? EM stands for electron, what? Microscopy. Electron microscopy, okay? And guys, because thankfully to that instrument, it has shown that the cytoplasm consists of a cytosol. Guys, what is cytosol? It is the fluid matrix, right? Then you have your organelles, the little mini organs. You have rough ER. What does RER stand for? Rough what? Endoplasmic, very good. Reticulum. Why do they call it rough? What does it have attached to it? No? Ribosome. Okay, it has a bunch of ribosomes. Rough endoplasmic reticulum. They call it that because it has a bunch of little ribosomes attached to it. Okay? The smooth ER doesn't have any, but the rough ER has a bunch of that. Guys, in the neuron, the RER is addressed or they're known as Nissl bodies, okay? Rough ER is a term we use for just general cells, but in the neurons, the Nissl body, or in the neurons, the rough ER is known as Nissl bodies in the neurons, okay? And also the cytoplasm has these so-called inclusion bodies. They're simply the non-living or just simply chemicals that are found in that. I'll go into more detail and I'll give you a little checklist of types of inclusion bodies that occur in neurons, okay? Then you have the Golgi complex, guys. That's the pretty much the traffic director. So whatever's produced, for cellular proteins, or for the Nissl substance, okay? Now the Nissl bodies forms arcs around the nucleus. I'll show you a picture of that in a bit, a little diagram, okay? The Golgi, it modifies, concentrates, and packages everything, guys. So whatever's produced from the Nissl substance, right? The Golgi, guys, think of the Golgi as like the UPS of the cell, okay? It has a receiving side and it has a shipping side, okay? So then pretty much the circulatory vesicles pinch off from the transphase and then it fuses to the membrane. So let me show you a diagram that actually, maps this out better than just talking about it, okay? So look here, guys. See this picture right here? Okay, so it shows you, let me do this. So guys, here's your rough ER, right? So that's your endoplasmic reticulum. Has all these little circles. This represents the what? Little ribosomes attached to it, right? So in the neuron, we call the rough ER the Nissl substance, right? So notice how it pinches off here. Okay, pinches off, and then you have the Golgi. You have the receiving side and the what? And the shipping side, right? So whatever's produced from the Nissl substance, then it goes here, and then it works its way towards the plasma membrane of the neuron, or I should say, in the case of the neuron, it will work its way all the way down to the terminus of that axon and release the neurotransmitter, okay? So in the case of the neuron, think of the Golgi as the part that produces the neurotransmitter, or I should say, the little vesicles that carry the neurotransmitter pinches off of the Golgi, okay? That's how those little vesicles are formed. Okay, what else, guys? Then you have your mitochondria. That's considered the powerhouse of the cell, right? It's a double membrane organelle. The inner membranes form these little enfoldings called cristae. It is here, guys, where the ATP is produced, okay? Has its own DNA and RNA, and it's involved in cell respiration. Now, guys, what are the 3 steps with regards to cell respiration? First step is very good, glycolysis, guys. Okay, very good. Next one is the Krebs cycle, very good, Krebs cycle. Third one is electron transport chain, very good. Guys, real quick question for those that saw the video. Where during which phase is the most ATP harvested? Here, exactly. It is here where most of the ATP is harvested, okay? What is glycolysis? This is where the, what? Glucose is what? Split into two what? Two pyruvates, very good. And then guys, it is in the Krebs cycle that what? That where you have all those compounds. Now so guys, here's a picture showing you, again guys, I have a video that actually walks you through, step by step of the Krebs cycle. So here's the outer membrane, here's the inner membrane of that mitochondria, and then the little, shapes, spaces are the cristae, right? And then guys, and notice how you have, if you zoom in on the little inner membrane of that, of the mitochondria, you'll see the little enzymes that are involved in the production of ATP. So guys, so let's name some of so you have your two pyruvates, right, that are products of glycolysis. Now, where does glycolysis occur? Where? In the cytoplasm, right, guys? In the cytoplasm of the cell, that's where glycolysis occurs, okay? What breaks down did the video talk about did it mention this, guys? Hexokinase? Did it mention that? Okay, that's the enzyme that breaks down the glucose into two what? Two pyruvates, okay? So just watch the video, check that out. So then, guys, when we're now in the Krebs cycle, we can name some of the compounds, right? So in the Krebs cycle, you have, what's the first one? Citrate. The second one is what, guys? Isocitrate. You guys don't have to write this down, okay? It's in the video. I mean, but if you want to write it down, go ahead. Then you have, what's next? Alpha, ketoglutarate. Is that right, guys? Okay. What's next? Succinyl CoA, right? What's next, guys? Succinate. What's next? Fumarate. What's next? Malate. What's next? Oxaloacetate, right? Exactly. Okay, 1, 2, 3, 4, 5, 6, 7, 8. There's 8. Okay, remember, and it forms a loop, okay? Now, guys, what's key here is that you know how many what? It's all about how many what? How many carbons. So how many carbons are in citrate? How many in isocitrate? How many are in alpha-ketoglutarate? 5. Then we have what? 4, then what? 4, then 4, then 4, then 4, then what? Then 4. Is that right? Then it gets 2 from the what? Acetyl CoA. So 4 plus 2 is what? That's how you get that. Okay? So guys, the whole key here is that what I want you to focus on is make sure you know how many what? Just how many carbons are in these compounds, okay? It's all about just knowing how many carbons, guys, okay? Just 6, 6, 6, 6. It's 6, 6, and then you go 5, 4, 4, 4, 4, 4, 4, and then you get 2. Okay? That's it. So make sure you watch the video so you know what's going on, guys, and you're good to go. And guys, bottom line, again, where do you harvest the most ATP? Electron, that's where most of the ATPs are harvested, in the electron transport chain, okay? How many total ATPs are produced, guys, how many total ATPs are produced? Well, first of all, cell respiration is simply the breakdown of glucose into simpler what? ATP. That's the whole summary of cell respiration. Breakdown of glucose into simple ATP, that's what fuels the cell. How many total ATPs are produced per glucose molecule? Total ATPs? 36 total, okay? But again, guys, just watch the video and you'll get a little mapping of that, okay? All right, guys, moving forward. Let's get out of this. Okay, then you have your cytoskeleton, okay? Your cytoskeleton, guys, that's what supports the cell's structures, right? Provides the machinery for, various cell movements. And guys, and here's some of the components. You have your neurofibrils, neurofilaments, microfilaments, and microtubules, right? Now, not much to add to this, guys. You have your microfilaments, the thinnest elements of the cytoskeleton. They form a little network right underneath the membrane, guys. So think of the microfilaments as like a glue. It's like a glue that adheres the plasma membrane to the underlying cytoplasm. Okay, think of it as like a little glue. Okay, then your microtubules are the elements with the largest diameter. They are made up of these protein subunits. Guys, then you have these motor proteins. You have kinins and you have dyneins. So there's a little video that kind of just gives you a little overview of that. Very brief video, like 5 minutes in duration, okay? And guys, what these motor proteins do, they simply what? Move the organelle along the what? Microtubule, okay? So guys, think of the organelle, okay, think of this organelle as like, I don't know, let's call this organelle, let's call this a vesicle that has what? A neurotransmitter, exactly, neurotransmitter. And guys, those things move along from the body of the soma, right, body of the neuron towards the terminus. They travel along these what? These microtubules, right, via these motor proteins. Now, did the video talk about kinesins? Okay, now kinesins move organelles from what? From soma to terminus, is that right? So it moves, so guys, what direction is that? Anterograde, okay? So pretty much anterograde, remember, it's from the soma to the terminals, okay? So the kinesins are what moves stuff in those directions. That's the motor protein that moves it from the soma to the terminal. What's the other protein? Dyneins, okay, dyneins move the stuff from the terminus, right, back to the soma. Very good, okay? Guys, just think of the little motor proteins as like little feet, okay, that travel along, that move along the microtubule, okay, the microtubules inside the neuron, okay? All right, so kinesins are involved in anterograde, dyneins in terms of what? What do you call that? From terminal to soma, what do you call that? Retrograde, okay, that's retrograde. Okay, anterograde from soma to terminus, and retros from terminus back to the soma, okay? Via these little feet called the motor protein. Okay, what else, guys? You have microtubules. Lysosomes, guys, where do they originate from? So, where do you find them? So, guys, if you go back to this picture, where do you think these lysosomes originate from? They come from where? From the Golgi, okay? From the Golgi. Think about it, guys. It pinches off here, and then you have what? You have a lysosome, okay? So they pinch off. Guys, and what lysosomes do, they simply just, they're little vehicles, little vesicles that carry what? Enzymes, hydrolytic enzymes to break stuff down. So once something is inside, let's say you have a worn out organelle, right, coming from the terminus back to the soma of the neuron, and then it's traveling in this, this little organelle here, and then you have a lysosome going to attach or fuse to that organelle, and then secrete its enzymes into that organelle to break it down. Okay? So they originate from the what? From the Golgi. They originate from the Golgi, so they pinch off from the Golgi apparatus, okay? And here's their function, to degrade worn out or non-functional, organelles and so forth, okay? What else, guys? So moving forward, then you have your centrioles. They're small, they're barrel-shaped organelles. They're right angles to each other, and they are actually 9 triplets of microtubules. Okay, they're actually arranged in terms of 9 triplets of microtubules. Okay. Not much to add to that, guys. Centrioles, and then you have here you go. Here are the inclusion bodies. Now, guys, these inclusion bodies are pretty much non-functional units. They're just simply chemical substances that occur in neurons, in cells in general, but in neurons. I focus on the neuron. So, let me give you a little checklist of examples of inclusion bodies that can occur that can occur in neurons. You have lipofuscin. Okay, this is an inclusion body that accumulates. You accumulate lipofuscin as you increase in age. So the older you get, over time you start to accumulate lipofuscin. Okay, they're simply very telltale characteristic of older folks, right? You can actually pull out if you observe a neuron, and if it has a good amount of lipofuscin, most likely it is an older individual. So they actually accumulate over time or as you get older. Okay. Another type of inclusion body includes melanin. Okay, now melanin, you find this in what's known in a part of the nervous system known as the substantia nigra. Okay, Guys, this part of the nervous system, of the CNS, is responsible for, or I should say of the midbrain, is responsible for the production of a neurotransmitter known as what, guys? Dopamine. Okay, this part of the nervous system produces dopamine. Okay, so melanin is evident, is what is most evident in that part known as a substantia nigra. Okay, which is responsible for the production of dopamine. If you start to have a reduction in the number of melanin, that's most likely that person is most likely a candidate of having what? Parkinson's. Okay, of having Parkinson's disease. All right, what else? You have what's known as Negri bodies. Okay, this is a telltale of a person that has what? That got infected with rabies. Okay, you have Negri bodies. You have what's known as what's the other one? Lewy bodies. Okay, this is indicative of a person that has Parkinson's. If you find Lewy bodies in a person's neurons, most likely they have Parkinson's. Okay. You have another one known as Hirano bodies. Hirano, Hirano, Hirano bodies. Indicative of a person that has Alzheimer's. Right, guys? And that coincides with what person's what? Memory, right? Alzheimer's, okay. What's the other one? Cowdrey type A. This is indicative of a person that has herpes simplex encephalitis. Okay. What else? Tangles. I'll just stop there. So guys, so here's some examples. So okay, guys, here's a little checklist of some inclusion bodies that can occur that you can find in some of these neurons. So this is normal. This is not a big deal here. So this occurs as you get older. However, you know, this is normal in the substantia nigra that you learn about this when we get to the parts of the brain. The midbrain is the part that's responsible for producing dopamine. Obviously, if you have a reduction in the number of these, person probably is going to develop eventually Parkinson's. Negri bodies, person has rabies. Lewy bodies, person has Parkinson's. Hirano bodies, person starts to develop Alzheimer's. Cowdrey type A, person has herpes simplex encephalitis. Okay, so make sure that you are aware of these inclusion bodies that can occur in some of these neurons. Okay, guys, then you have your cell membrane. Now, guys, what the membrane does is separates two environments. Separates the extracellular from the what? Intracellular. Okay, separates outside from inside. Regulates the exchange of stuff coming in and out. In terms of the sensitivity of the membrane, it can be affected by changes in the extracellular fluid. All right, guys, you have what's known as ECF. And you have what versus what? ICF. What does ECF stand for? Extracellular fluid, and then you have fluid where? On the inside of the cell, okay? So, and then you have receptors for recognition and communication, guys. I think I talked about that earlier, but receptors that you find on the membranes. Like, for example, you saw the case of receptors on the postsynaptic membrane, right? Okay? Now, obviously, alterations can affect the function, okay, its function. It's also the plasma membrane is involved in simply for structural support, okay? All right, now let's talk about very quickly, guys, membrane potentials. Now you have what's called depolarization. Here are a couple of events: depolarization, hyperpolarization, right? And you have action potentials. So, guys, what happens with regards to these potentials is that in the case of depolarization, the inside of the membrane of the axon becomes less negative, and in hyperpolarization, the inside becomes more negative, right? And then you have what's known as what are known as action potentials. That's the initiate. This is the part that initiates. I should say the AP is what the part that coincides with the nerve impulse. This coincides with the so-called nerve impulse with the signaling. Okay, the signal. I'll talk about that in a bit, guys. So you have depolarization, the inside becomes less negative, so forth. So let's talk about that real quick, guys. Now, guys, with regards to generating an AP, what does AP stand for? Action potential. Okay, to generate an action potential, you have these phases. You have the resting stage, the depolarizing phase, and the repolarizing phase, okay? So during the resting phase, folks, just make sure that you're aware that during the resting stage, the voltage-gated channels are closed, okay? Or it's safe to say that the insides were negative, right? I'll show you a picture of that. When it starts to depolarize, when the memory starts to depolarize, the permeability of Na, what does Na stand for, guys? Sodium, okay? It becomes more, there's a permeability of sodium, right? And when it repolarizes, during this phase, what's this K? Potassium. There's a permeability of potassium increases. So guys, let me just jump to the picture. Let me cut to the chase here, guys. So look at this picture, guys. This picture pretty much summarizes what happens during all this stuff of generation of an action potential, okay? Look here, guys. So this is a voltmeter right here, a voltmeter, okay? So at rest, guys, notice how the inside of the axon is more what? At rest is more what? It's more negative, okay? So at rest, we're talking about minus 70, what does MV stand for? Millivolts. Okay, millivolts, okay? At rest, bless you, it measures at 0.80. So right now, at this point, there's no signaling, no action potential, okay? So it is minus 80. Other sources may say minus 80 or minus 70. Well, either one's correct, okay? But bottom line here, it is at rest. It is resting, okay? At rest, at minus 80. Now, here, notice how it is becoming what? Less negative, right? Less negative is moving in which direction, guys? Towards what? Towards positive. It's going to reach this point, positive at plus 40 millivolts, okay? So this is known as what? Depolarization, guys. And what's happening here is you're going to increase the permeability of what? Of sodium. So what's going to come in? You're going to have an influx of what? Influx of what, guys? Of sodium. Okay, you have an influx of sodium, right? When it depolarizes, right? And then here, notice how then it reaches threshold, and then it's going to go back down to rest. But at this phase, folks, what's going on here? It's more permeable to what now? Potassium. And here, you're going to have a what? An efflux of what, guys? Of potassium leaving going from the inside out, okay? And at this point, notice how the inside of the axon is becoming more what again, guys? More what? Becoming more negative. So it's going back down, becoming more negative. It's going back down to rest, okay? So you have at rest, you have depolarization. You have what here, guys? This is known as what? Repolarization, okay? Going back down to rest, okay, guys? So this pretty much explains, maps out what happens during what, guys? During a what? An action potential, okay? You have at rest, then it becomes, at rest it is more negative on the inside, right? And then once it starts to depolarize, it becomes less negative, right? Moving towards plus 40, then then you have an influx of what? The channel for sodium open up, and then when it repolarizes, it becomes permeable to what? Potassium goes out, okay? This is simply showing you what happens when you have multiple signals. This is known as summation, okay? Just summation, that's all. Summation, you have several of these potentials, all sub-thresholds, so forth, but then you have a bunch of them, then you have summation, okay? But that's all, I'm gonna add to that. Bottom line, folks, that just make sure you know what happens, that you have at rest, the inside is less negative, right? At rest, when it depolarizes, becomes less negative, becomes actually positive, right? Once it gets to 40, at this point you have depolarization, and you have influx of what? Sodium, and then when it repolarizes, becomes less negative again on the inside, and what comes out? What leaves? Potassium goes out, okay? Efflux of potassium, okay? Guys, then you have what's called the neuroglia. This is the support cast. So you have the main, we talked about the neuron, that's the main star, the main functional part of the neural tissue, right? Then you have the support cast, that's the supporting cells. You have 6 types. You have 4 in the CNS, 2 in the PNS, and they each have a specific function. Their function primarily is to scaffold neurons. Their chemical production guides young neurons to proper connections, and they promote the growth and health of the neuron, okay? Now guys, as far as the neuroglia of the CNS, they include the following. You have astrocytes, you have what? Microglia, you have what? Epidermal cells, and you have what? Oligodendrocytes. Now, what I want to add to this very quickly, folks, is the following. Two of these neuroglia, astrocytes and oligodendrocytes, they can be considered deterrents. Deterrents for axonal regeneration. Where, guys? In the CNS. Okay? So guys, bottom line, if you sever an axon in the central nervous system, what happens? That's it. There's no connection. You cannot regenerate that. Okay? I'm talking about the CNS, guys, not peripheral. The CNS. Okay? If you have a lesion, a cut in a nerve fiber in the CNS, you cannot regenerate that. And the end product is what? You're what? Paralyzed, right? Okay? So now astrocytes, guys, they are the most numerous and versatile of the glia. They have specialized. They have arms that radiate. Right? They anchor neurons to capillaries. By anchoring to neurons, right, or anchoring the neurons to capillaries, they form BBB. What does it stand for? Blood-brain barrier. Okay, it forms a blood-brain barrier, pretty much. Okay. They control chemicals, i.e. potassium, or just simply recycling of neurotransmitters, right? Now the microglia, guys, these are the cleanup crew. They keep the neural tissue clean, pretty much, okay? Epidermal cells, these are a type of a simple squamous epithelium. Okay, I should say they are a type of specialized simple epithelium, okay? Talked about that, I think, last class about basic tissues, right? They can range in shape, they can be squamous, or they can be columnar. They line the central cavities of the CNS, and they circulate CSF. What is CSF, guys? Cerebral spinal fluid, okay? They circulate cerebral spinal fluid. You'll learn more about how CSF is produced when you tie in epidermal cells with a network of capillaries, okay? But you'll learn about that eventually in the course. And then, guys, your oligodendrocytes are simply producers of myelin sheaths of the axons in the what? In the CNS, okay? You call them Schwann cells in the PNS, but in the CNS you call them oligodendrocytes, okay? So you have astrocytes, microglia, epidermal cells, oligodendrocytes. Those are the four types of glia cells in the CNS, okay? Now, you guys, here's a picture showing you the cast of characters, guys. Here's an oligodendrocyte. Notice how it's what? Producing myelin sheaths, right? Around the axons, okay? Here are the microglia right here, cleanup crew. Here's the astrocyte. Notice how it's embracing a capillary, right? And notice these little feet surround the capillary, forming the what? Blood-brain barrier, okay? And guys, here are the epidermal cells lining the ventricle of our brain. And this circulates what? Circulates what? CSF, okay? So, and then guys, here's a picture. Oh, that's right. Here's a picture showing you two types of astrocytes. Here's a protoplasmic. And here's a fibrous astrocyte. Okay, here's a fibrous astrocyte. Fibrous astrocyte, here's a protoplasmic one. Guys, the protoplasmic astrocytes, you find it in gray matter, right? You find it in the gray matter. You'll find the fibrous one in the white matter, okay? You'll learn more about this white matter, gray matter eventually in the course, guys, but just stick to this for now, okay? So you find this here and here. Guys, what I want to emphasize on these astrocytes is think in terms of, well, let me go back to this real quick, guys. What happens when a neuron dies? When a neuron dies, right, in the CNS, do you get a new neuron? No, you don't. When a neuron dies, it is replaced by what? It is replaced by what? By an astrocyte. And that's what contributes to the what scar tissuing in the CNS, okay? That's why, like I said, that's why the astrocytes and this are considered deterrents of what? Regeneration of axons, where? In the CNS, okay? So if a neuron dies, you don't get a new one, it gets replaced by an astrocyte that really does not help in terms of regenerating the axon, okay, in the CNS. And the oligodendrocytes, they simply release compounds that do not help in the regeneration of the axon also. Now guys, back to this. So the reason why I bring this up is because these astrocytes, ever heard of this, guys? Gliomas? What's a glioma? That's a tumor. That's a tumor that occurs in the nervous system, okay, in neural tissue. Okay, gliomas most likely are arise from fibrous astrocytes, okay? Whenever you hear a person having a glioma, that's a person having a tumor in their brain, in their spinal cord or whatnot, okay, most likely that tumor is derived or arose from a fibrous astrocyte. Not a protoplasmic, fibrous astrocyte, okay? Glioma, that's a fancy term that describes a tumor in a person's nervous system, okay? Glioma. Let me see, guys, what do I have to add to this? You have your glia, that's astrocytes, microglia, vitamin cells, dendrocytes, okay? You have a slide that talks about Schwann cells and what do you call it? Emphysites?
There's the last one.
Satellite? The last slide? Okay, hang on, thanks. So, Warren, hang on. So,
No?
no?
My bad.
That's on what? Okay, so that's another set of slides? Okay, all right, I'll leave it. Cool, all right, excellent, thank you. All right, guys, so hang on. Excellent, great, good news. Okay, so folks, that ends my discussion. Okay, wait, hang on, guys, wait, hold on, sorry about that. Let me go back to the last slide. Hang on, sorry about that, hang on, real quick. So, okay, here we go. So guys, here's the picture, let me see what this says, just not to leave you hanging, guys, hang on, not to leave you hanging. So here's the picture showing you, here. So here's picture showing you two types of astrocytes. See right here, guys? These are the, look at these are the fibrous astrocytes, and these are the more thicker looking what? Protoplasmic astrocytes. Okay, protoplasmic astrocytes, okay? All right, so that looks good, folks, and I think we'll, now that officially ends my discussion. Okay, folks, so guys, listen up. So you have the answers, you have the information to the test right here, guys, okay? So you have your information here, so make sure, guys, that you transcribe all this information tonight. You have a couple of videos to watch. If you have to watch, make sure you watch the video on the Krebs cycle if you need that. But guys, I already told you what you want to focus on that. Just make sure you know everything about the Krebs cycle on the cell restoration. But what I want you to know also is number of carbon. Make sure you know number of carbon, etc. I already addressed that. And also, you know, kinesis, dyneins, you know, with that very short video. And then you also have what else? Video on peripheral nervous system. Is that right? Yeah. Peripheral nerve, and also on spinal cord. Okay. So you're good to go, folks. So you have all the answers to the test. All the answers, okay, folks? Have a nice weekend. Toodles. Make sure you work on the stuff tonight, folks. Don't fall behind, and you're good to go.